Hello, my name is Mohammed Noor, and I am a professor at Duke University. I'm very excited to be talking with you about genetics and evolution. During this first video, we'll be looking at what is evolution, and in some of the subsequent videos, we'll look at the evidence for evolution, and we'll look at responses to criticisms of evolutionary theory. So let's dive right in. Evolution, in a biological sense, is simply change through time. And very importantly, that is change through time over generations. So, for example, the human species hundreds or thousands of years ago was much shorter than it is today. Over the course of those generations, the human species has grown taller. That change is an evolutionary change. In contrast, I used to be a baby, now I'm an adult. That change is not an evolutionary change because it happened within an individual. That change is development instead. Now, one common source of confusion is the distinction between evolution and natural selection. Evolution, as I mentioned, is simply change through time. Natural selection is one driving force of evolution. It can be one reason that evolutionary change happens. But importantly, it is not the only reason evolutionary change happens. Later in the course, we'll talk about other forces that drive evolutionary change. But let's start by looking at one famous case of evolution by natural selection. A classic example of evolution by natural selection is that of the peppered moth, Biston betularia. I have here a picture of two of these. There's one peppered-looking one that's mostly white with black flecks, one black one over here, often referred to as melanic. Which of those is harder to see on that tree? Probably for most of you, the peppered form is a little bit harder to see because it blends in with the color of the tree and the spotting pattern of the lichens that are growing on the tree. In contrast, this black one is very easy to spot. Not only is it easy to spot for you, but it's also easy to spot for hungry birds looking for a tasty moth treat. As a result of this, the black forms tended to get eaten much more than the white forms, and in 1848, nearly all the moths that lived tended to be peppered. Now, things changed with the Industrial Revolution. The trees were blackened and a lot of the lichens died. So instead of having this form be the camouflaged form, all of a sudden the black form had the advantage on these new black trees. Because of this change, 98% of the surviving moths were now black, and the peppered forms were largely gone. This was not a change in the color of individual moths. What happened is the offspring of the black moths were able to survive because they could hide. They were better camouflaged. Meanwhile, the offspring of the peppered moths were not so lucky, and they got eaten up by a lot of the birds. So this change from being nearly all peppered to nearly all black in this species is referred to as industrial melanism. That is change in the coloration of a species to a darker form in response to pollution in the environment caused by humans. This happened not just in the peppered moth, but in a wide array of insects and other species. This process continued until about 1952. In 1952, in Great Britain, we had what was referred to as the Great Smog. The Great Smog brought in a ton of smog, as you would guess, that sat over the city of London for weeks on end. Tons of people died from this. This was a terrible amount of pollution. This picture is taken in broad daylight. It was incredibly hard to see. Ambulances didn't run. And in fact, it was even seeping into homes and performance venues. A lot of theaters closed because even inside the theater, so much smog had come in you could no longer see the stage. As is typical with a lot of politicians, it took a disaster before they could actually act. And what we had in Great Britain was the passage of the Clean Air Acts in 1956 and 1968. The passage of these acts cleaned up a lot of the environment. What you saw happening then was those trees that had previously been blackened were able to get cleaned off, and a lot of the lichens on them started to grow back. In response to this change in the environment, we also had a change in the peppered moth. So this was a case of evolution from white to black, and now from black back to white. This picture shows the change in the fraction of moths that were black over time. So starting in 1959, again, most of them were black, as we just said. So we go into the 1980s, we see a drop in the fraction of these black moths. 1990s, further drop. And in the 2000s, it's actually hard now to find a black form of this peppered moth in Great Britain, in the United States, in Japan. The same process happened on multiple continents. So this is a case of evolution by natural selection. So one question a lot of people have asked me is, isn't evolution by natural selection just a theory? Well, I will argue to you that evolution by natural selection is a mathematical inevitability. There is no way to avoid having evolution by natural selection if some very simple conditions are met. So let's imagine you're working with a population of squirrels. I used to live in Louisiana. Every April, we'd have an explosion of baby squirrels all over the place. There'd be baby squirrels running around in the yards, on the sidewalks, and kind of unfortunately for some of them, out into the streets, where they'd often get hit by cars, sometimes by me. This was very unfortunate for the squirrels, as you can imagine. Now let's imagine that you're looking at a population of squirrels of two types. Let's say there's a type A that runs randomly and is likely to run out into the street, or possibly, not running into the street. And let's imagine there's also a type B that fears asphalt, so they avoid going into the streets. There would be an advantage to the type B offspring because they're much less likely to get hit by a car if they don't go out into the street. So let's say that on average, the type A squirrels have one offspring and then they pass away, whereas the type B squirrels have on average two offspring before they pass away. So we'll start a population with half type A, half type B, and let's see what happens. And we're assuming that type A individuals have type A kids, type B individuals have type B kids. After one generation, these type A individuals each have one offspring, so we have now a new population of 100 type A squirrels. The B ones, as I said, are each having two offspring, so these 100 produce 200 kids. So what's happened? Instead of having 50% B, all of a sudden now we're 66% B. It's 200 out of 300. So let's go another generation. Well, these 100 have another 100 for babies. These 200 would have how many? They'd have 400 because, again, each of those 200 has two babies. We're assuming, again, the parents pass away. So we've gone from 66% to 80%. 400 out of the total of 500. Finally, generation 3, 100. And over here, we have 800 squirrels. So we've gone from 80% now to 89%. That is evolution by natural. Natural selection. We see this change over time, over generations, very importantly, from 50% type B to 89% type B over the course of 3 generations. Three new generations. Explosion of I will argue the conditions. There'd be baby squirrels running around working with a population of squirrels. I used to live in Louisiana. Every April, we'd have an explosion of baby squirrels all over the place. There'd be baby squirrels running around in the yards, on the sidewalks, and kind of unfortunately for some of them, out into the streets, where they'd often get hit by cars, sometimes by me. This was very unfortunate for the squirrels, as you can imagine. Now let's imagine that you're looking at a population of squirrels of two types. Let's say there's a type A that runs randomly and is likely to run out into the street, or possibly, not running into the street. And let's imagine there's also a type B that fears asphalt, so they avoid going into the streets. There would be an advantage to the type B offspring because they're much less likely to get hit by a car if they don't go out into the street. So let's say that on average, the type A squirrels have one offspring and then they pass away, whereas the type B is less likely to get hit by a car if they don't go out into the street. So let's say that on average, the type A squirrels have one offspring and then they pass away, whereas the type B squirrels have on average two offspring before they pass away. So we'll start a population with half type A, half type B, and let's see what happens. And we're assuming that type A individuals have type A kids, type B individuals have type B kids. After one generation, these type A individuals each have one offspring, so we have now a new population of 100 type A squirrels. The B ones, as I said, are each having two offspring, so these 100 produce 200 kids. So what's happened? Instead of having 50% B, all of a sudden now we're 66% B. It's 200 out of 300. So let's go another generation. Well, these 100 have another 100 for babies. These 200 would have how many? They'd have 400 because, again, each of those 200 has two babies. We're assuming, again, the parents pass away. So we've gone from 66% to 80%. 400 out of the total of 500. Finally, generation 3, 100. And over here, we have selection. I hope you enjoy the next generations, very importantly, from 50% type B to 89% type B over the course of 3 new generations. I hope you enjoy the next video. Hello, and welcome back to Introduction to Genetics and Evolution. In the previous videos, we talked about basic evolutionary principles and how evolution by natural selection is a mathematical inevitability. We also talked about how, despite this, there is some variation in how well people accept the truth of evolution and allow it to be taught in various schools. So in this lecture, we'll talk a little bit more detail about some of those basic principles of evolution, and we'll go into some of the evidence for evolution and common descent of species, common ancestry of species. So first, evolution has 2 fundamental processes. Okay? So you can have change within a lineage. So if you were to go far back in time, 60 million years ago, ancestors of the modern horse stood only about half a meter tall. Over the course of time, over the last 60 million years, you've had the evolution of the modern horse, which today stands on the order of almost 2 meters tall. So we have this change within a lineage. These pictures, by the way, don't necessarily depict actual horse ancestors, but they show fossils that were present at the same time as the presumed horse ancestors. Okay? The other fundamental process within evolution is the formation of new lineages, or this idea of common ancestry of species. So if you were to go back, many millions of years ago, horses, donkeys, and zebras all shared a single common ancestor species. But then they had the split of this single species into what became 3 new species. One of them evolved into the modern horse, one evolved into the modern zebra, one evolved into the modern donkey. So from taking these 2 processes, there are 2 fundamental principles of evolution that I'd like to emphasize. The first is that much of evolutionary change, much of what's happening within a single lineage, is caused by natural selection. And it's this natural selection that produces the appearance of design that I talked about in a previous video as being something that sometimes causes confusion for people and misunderstanding. The other thing is that all species, not just a subset, all species share common ancestry. That not only do horses and donkeys have a common ancestor, but horses, donkeys, and pigs have a common ancestor. Horses, donkeys, pigs, and mice have a common ancestor. We share a common ancestor with them. Flies share a common ancestor with us. Coral shares a common ancestor with us. Sunflowers share a common ancestor with us. And even the amoeba shares a common ancestor with us. There is a presumed single origin of life that then produces all the diversity that we see on the planet today. We'll talk about that a little bit more extensively in a later video. But let's go from this into some predictions that come from this idea that most evolutionary change comes from natural selection and from the presumed common ancestor of species. Some of these we've already touched on in previous points. First, if life originated on Earth a single time in the distant past, then what we should see is when we go way back in the fossil record, the earliest detectable forms of life should be very primitive, should be very, very simple. And only later should you see some modern forms. Now it's possible that later you'll also see some simple forms, but the earliest form should certainly be simple. There's a famous quote from the biologist J.B.S. Haldane that what observation would refute the truth of evolution and common ancestry. And he argued that what would do it would be finding fossil rabbits in the Precambrian era. So this is a very, very long time ago, finding fossil rabbits. That would refute this idea of common ancestry. Well, that's never been found, so there has not been an observation to refute the truth of evolution or common ancestry. Well, in fact, when you look at the fossil record, the early life really is extraordinarily simple. You see things which are either single-celled or very, very small and with very simple structures very early in the fossil record. And as you go later in there, you start to see the earliest simple-shelled animals. And much later do you start seeing things like plants or vertebrates. And only very recently you see humans. So in general, yes, the earliest life we see evidence for is extraordinarily simple. We don't see these ancient vertebrates that are arising long before simple gastropods or other shelled animals. So that's one prediction fulfilled. Here's another one. If evolution occurred within lineages and those lineages sometimes split, we should be able to find cases of species gradually changing the fossil record from one form into another form. And in fact, I've actually already shown you the answer to this in the case of horse evolution. When we look at ancient horse forms going back in time, we see all these sorts of transitions there. We see this gradual change over time in the shape of the horse from the ancient horse to the modern horse. We see this not just in overall form, but we see this in various of their skeletal structures. So looking at, for example, their legs. The earliest horses there didn't have hooves, but had these individual toes. And you see this change over time towards the modern hoof. Same sorts of things in terms of their molars. So again, we see this change over time as predicted from common ancestry and evolution. Another prediction for answer should be able to find cases of species gradually changing as positional fossils from common ancestry and evolution. Another prediction we have is that if all creatures share a common ancestor, we should see transitional fossils, things that connect modern groups together. Now, one case which would be very interesting in this context is the case of birds. Birds are actually glorified reptiles. If you look at how birds are related to all the animals, birds are actually nested within reptiles. So there are some reptiles that are actually more closely related to birds than they are to other reptiles. So what we should see then is some sort of transition from this ancient reptile, which should have some characteristics of modern reptiles, to the modern birds. So we should see things like the evolution of feathers, we should see things that predate the beak, etc. In fact, that's exactly what we see. We see feathered dinosaurs that existed long before today. Right, so this is an example of Sinornithosaurus millenniae. And if you look at it, it clearly does have feathers in there. We can see this in the fossil. Notice it does not have a true beak, but in fact it has a mouth with teeth, even though it has a shape kind of similar to a beak. So this confirms this. But importantly, this also appears at the right time in the fossil record. This is found in deeper strata within the ground than modern day birds. This is one artist's conception of what this animal may have looked like. It's a very colorful idea for it. Now, critics of evolutionary theory often point to this idea of quote-unquote missing links. They say, oh, there's all sorts of missing links in the fossil record. And one kind of ridiculous example, and I think they would confess this is a ridiculous example too, that they give is that if you look at the modern crocodile and the modern duck, we don't see the transitional fossil, which you might expect to be a crocoduck. That's just silly. Nobody expects there to be a crocoduck. The reason here is there's some misunderstanding. We expect transitions between ancient forms and modern forms. We don't expect things that are connecting modern day forms to each other. That would be more like a hybrid rather than an evolutionary change. So let's look at this in the context of the evolution of cetaceans. Cetaceans are these large marine mammals like dolphins, porpoises, whales, etc. Now these are thought to evolve. These are mammals. They breathe air. They're thought to have evolved from land-dwelling mammals. So, they would share an ancient common ancestor with, for example, the hippopotamus, as you see in this picture. Now, if you go back in time, back to, say, 1970, so not very far back in time, there were not a lot of fossil connections between the cetaceans that we see, those living water-dwelling mammals, and land-dwelling mammals. We have one example here, the Dorudon. Well, again, as we started going through the fossil record between 1970 and today, more and more transitional fossils were found. So I'll click through a few of these quickly. You don't have to know them. You don't have to know their exact names, but just to illustrate the point of finding these missing links over time. 1979, we have this form found. 1993, another transitional form. 2000, another transitional form. 2001, another transitional form. 2007, another transitional form. Now, importantly, these actually really fill in a lot of the gaps that we see in the evolution of modern cetaceans. Some of these forms, such as this one right here, were found around 40 million years ago. A lot of these other forms date back to about 50 million years ago. So we see eventually these forms that have characteristics of land-dwelling mammals, but also have some of the characteristics of these aquatic mammals. And they're found at ancient times in the fossil record around the time you would expect these evolutionary changes to have happened. So there are not actually so many missing links as people would have you believe. There are some still missing, but that doesn't mean they don't exist. That's a very important point. So I talked so far about a few predictions of common ancestry and evolution over time, driven especially by natural selection. Let's change gears a little bit, and let me talk about some retrodictions. So these are observations that support the idea of evolution and common descent, but they won't necessarily have been things that we predicted ahead of time. That when we see them, they support this idea, but if we didn't see them, it doesn't necessarily mean that they don't support it. So these are things that are consistent with the idea of evolution by natural selection and common ancestry of species. One great example coming from this idea of cetaceans coming from land-dwelling mammals is vestigial organ, and specifically the vestigial hind limb bones of a gray whale. Gray whales don't have legs sticking out of their back, yet we see these hind limb bones there. This is very difficult to explain, except in the context of common ancestry, that ancient forms related to the modern whales actually did have these hind limbs. And over time they lost the hind limbs, but a couple of the bones have actually persisted over time. So this is a vestigial organ. It serves no known function in the modern gray whale, but it is something indicative of its evolutionary history. Here, by the way, is a picture of Jerry Coyne pointing at the hind limbs at a museum in Harvard. By the way, I should point out a lot of the examples I'm giving here are taken from a presentation by Jerry Coyne and are discussed in his book, Why Evolution is True. Another example related to vestigial organs is vestigial genes. These are genes that we see in our genome or the genomes of other species, but they don't work. They actually have some sort of disruption, so they don't actually produce the proteins or the structures that they do in other taxa when they're normally functioning. So one example here are the genes for making yolk proteins in mammals. So mammals, as you know, don't produce actual yolk. They produce this empty sac that appears in the embryo and doesn't have vitellogenin. So this is not something that's useful at all. Yet, the genes for making these yolk proteins are actually found in mammalian genomes. Why would this be? Again, it makes sense in the context of common ancestry that some of our ancient ancestors actually did make these yolk proteins. When we stopped needing them, they stopped making the protein, but the gene is still there, some remnant for it. The genes for making vitamin C are also not found in the genes for making vitamin C are found in primates, but we don't make vitamin C. If we don't take vitamin C, we get the disease called scurvy. We have to get vitamin C from our diet. But most other mammals, except for guinea pigs, actually produce their own vitamin C. Now, we have the genes for making vitamin C, but they're broken. So why would this be? Again, it's indicative of this change over time, that we have ancestors that actually did make their own vitamin C, and then there was an evolutionary change that made us stop doing it. The last one I'll mention is olfactory receptor proteins, that we primates are definitely lacking a lot of olfactory proteins that a lot of other mammals have. We can't smell nearly as well as a lot of other mammals. Yet, those proteins, the genes for making them, are still found in our genomes. They just don't work. Let me talk about another set of retrodictions. These are related to biogeography, basically where animals are found or where plants are found in the world. Well, what we see in terms of biogeography is that when you look at very isolated oceanic islands, they tend to have many native birds and insects, but they lack amphibians, mammals, or freshwater fish that are native to those islands. Now, why would this be? Now, if life just spontaneously arose in multiple places, you'd think it would be just as likely to arise spontaneously in a continental area as in an oceanic area. But you don't see these particular forms in these very isolated areas. Well, there's two explanations. One is that life spread across these continents, and the only forms that are found on these isolated oceanic islands are the ones that could get there, right? The ones that were able to fly over there. The alternative hypothesis is that mammals or amphibians can't live well in these oceanic islands. Well, we can test, and we have in fact tested unintentionally, the latter hypothesis. Mammals and amphibians have been introduced to isolated oceanic islands, and they do extremely well sometimes. Here are two examples from Hawaii. The cane toad, which was also introduced to Australia, which is both a continent and a very large form, where animal genomes found or where plants are found in the world. Well, what we see in terms of biogeography is that when you look at very isolated oceanic islands, they tend to have many native birds and insects, but they lack amphibians, mammals, or freshwater fish that are native to those islands. Now, why would this be? Now, if life just spontaneously arose in multiple places, you'd think it would be just as likely to arise spontaneously in a continental area as in an oceanic area. But you don't see these particular forms in these very isolated areas. Well, there's two explanations. One is that life spread across these continents, and the only forms that are found on these isolated oceanic islands are the ones that could get there, right? The ones that were able to fly over there. The alternative hypothesis is that mammals or amphibians can't live well in these oceanic islands. Well, we can test, and we have in fact tested unintentionally, the latter hypothesis. Mammals and amphibians have been introduced to isolated oceanic islands, and they do extremely well sometimes. Here are two examples from Hawaii. The cane toad, which was also introduced to Australia, which is both a continent and a very large island, it has done extraordinarily well there. This is an amphibian. It went in there. It's destroyed tons of the native fauna that are present on these islands. So clearly, it's okay. The Indian mongoose shows an example of a mammal that was introduced to Hawaii, and again, killed off a lot of the native fauna, and it's a big pest species there. But they can live on these oceanic islands. So we hypothesize that the reason that they're not found on these islands is because they couldn't get there. Another retrodiction related to biogeography is that the most similar species to those you'd see on oceanic islands. So let's say you have some birds on an oceanic island, you should expect their nearest relatives, or the ones most similar to them, should be the ones on the nearest mainland. Again, indicating this idea that the animals that got to islands that became native species there over a long period of time are ones that were able to fly over there from the nearest continent. And in fact, you do see that if you look at isolated oceanic islands and the nearby continents, you tend to see very, very similar forms. In contrast, if you look at an isolated oceanic island, look on the other side of the Earth, you don't see things that are that closely related. The last retrodiction I'll mention, and I'll only talk about this one briefly, is that of inefficient design. That if an organism were to actually be designed from scratch, it wouldn't be designed the way it actually is. However, when you think of it in the context of being cobbled together over time from evolution, it does make a lot more sense. Now, one example that's often raised in this context is that of the laryngeal nerve that comes off the vagus nerve. Now, in fish, this branch off the vagus nerve, it has a pretty short little transit down to the gills. No problem at all. What's a little bit less efficient is when you look at humans or other mammals. So here's this laryngeal nerve loop, comes down, loops under the aorta, and then comes all the way back up to the larynx. So that's not very efficient. That's a long way for this to transit when it could just go boop, just connecting right across like that. It's even less efficient when you look at the giraffe. It starts way up here at the brain, goes all the way down, again looping around the aorta, comes all the way back up and connects over there. That's extraordinarily inefficient. If its primary goal is to actually drive the larynx, that really doesn't make sense. However, probably what happened is this nerve happened to be going around the aorta in an ancient form. There, it didn't really matter. It couldn't cut and reconnect around it over evolutionary time because every one of its descendants of this ancient animal needed to have a functional laryngeal nerve. So evolution was working with the materials available. It couldn't actually produce a more efficient design. You can think of it in the context of retrofitting a house without ever turning off the electricity or water. That there's so many things you'd have to do that would be very, very inefficient that if you just destroyed the house and built it again from scratch would be far more efficient. Well, this is the kind of thing we see in the context of species out there. In later videos, we'll be talking about responses to criticisms of evolutionary theory. I hope you'll join us. Thank you. Hello, and welcome back to Introduction to Genetics and Evolution. In the first video, we talked about why evolution by natural selection was mathematically inevitable. There was no way to avoid having evolution by natural selection if certain very simple conditions were met. We also talked about one famous example of evolution by natural selection, that of color change in the peppered moth in Great Britain and other parts of the world. Now, it may surprise you to learn after seeing that video that in many parts of the world, people don't believe in the truth of evolution. Let's talk a little bit about some of the historical challenges there and some of the issues that people are facing, particularly in the United States. Now, one of the most famous examples of opposition to evolution is the Scopes monkey trial from the 1920s. In the United States state of Tennessee, they passed a law called the Butler Act, which made teaching evolution in public schools unlawful. Now, one teacher challenged this law. His name was John Scopes. He went into his classroom, taught evolution in a very public forum. He was taken to court as a result of this, and in a very highly publicized trial, as you can see from some of these headlines, evolution trial opened by prayer, judge has a Bible. It brought out all the people who are against evolution on one side and for the teaching of evolution on the other side. Now, John Scopes did actually violate the law. As a result of that, he was actually found guilty. However, trivializing this law and showing how ridiculous it was, he was fined a mere $100, which was not that much money. So this was actually a big victory for the pro-teaching evolution community. Now, the prosecutor, the person who was basically trying to send him to jail, was William Jennings Bryan, who had previously run for president of the United States. He was extremely disappointed. He lost his chance to give a very impassioned speech against evolution. So this was a big victory. Now you may be thinking, well, this was 100 years ago. Does this kind of thing still actually happen today? The answer is yes, and not just in the United States either. Here are a few examples from just the last couple of years, long after the Scopes monkey trial, of course. The first one is from the United States state of Tennessee in Livingston Parish. Creation Science Genesis, school system looks into teaching anti-evolution. It's just from a few years ago. Even more recently, this is a case from outside the United States. South Korea surrenders to creationist demands. And very importantly, look at what they were saying. Publishers were set to remove examples of evolution from high school textbooks. So they actually have this in their standard curriculum. They're basically wanting to take it out, remove these examples from it. So there's a lot of public pressure to not talk about evolution in public schools. See the same thing in the United States state of Kansas, headed for another debate over evolution in 2005, just so you can see a couple of comparisons. So this was a survey done in 2005. The red bars indicate people who, when asked about the truth of evolution, said that evolution is, quote, Hello, and welcome back to Introduction to Genetics and Evolution. This is going to be the first video talking about genetics, or the patterns of inheritance. I'm very excited to go into this with you. Now, before we go into the details, there are a few basic terms I'd like you to know. If you're not familiar with these terms, I'd encourage you to use some of the resources that are linked from the online site to go learn more about them. So I'll introduce these terms very briefly because I'll be using them repeatedly throughout this video and other later videos. So the basic terms I'd like you to know are first that of DNA, that's short for deoxyribonucleic acid. DNA, as many of you know, is the hereditary material that is used by most species, and it's comprised of nucleotides, or building blocks. Those building blocks are labeled A, C, G, and T. So there's 4 different forms that are commonly used in DNA, and that's how they're often abbreviated, A, C, G, and T. Now, genes are the fundamental units of biological information, and they're made of DNA. Typically, a gene will be on the order of a couple of hundred or maybe a few thousand DNA nucleotides long. So these pieces put together to make this particular set of instruction. Okay? Chromosomes are organized packages of DNA. We humans, many of you probably know, have 23 pairs of chromosomes, the most famous pair being that of the X and Y, which determine our gender. Now, these packages bear genes along their length. These are very long stretches of DNA. They're millions of bases long typically, and they have genes along them. So basically, they're the scaffolding for genes within our body. And finally, the genome is the entire set of DNA instructions in a cell. So all the chromosomes and any other material like that is the genome, or you often hear reference to the human genome. So I hope you already appreciate this, but I'll say it nonetheless. Genetics is very cool, as illustrated by this handsome young man right here playing with genetics. So genetics is this code that's found inside every single cell of your body, right? And it affects everything about you. It affects how you feel, it affects how you learn, it affects how you look, it affects everything. It affects how long you'll live, it affects what diseases you have. I mean, it's just amazing how much is affected by genetics. Now, when I say affected, I don't mean that it's completely explained by genetics, but genetics has a big effect on everything about you, and not just about you, but also about your pets. You know, in fact, why are your pets the particular species they are? They probably came from an egg that wasn't too dissimilar from the egg that you came from, but they changed into, over time, this, you know, puppy or this kitten or something like that. Again, this is pretty cool. This is all within this code. Livestock, we can actually help better breed better cattle by understanding the genetic basis of traits. We can attack pests of our crops. We can attack parasites better within our body by understanding the various pieces of their genetics. And ultimately, and you'll see this later on in the course, we can understand evolution much better by having a good understanding of genetics. Now, poor Darwin was able to come up with the ideas of evolution by natural selection without having access to a lot of detailed genetics. But over time, in the last, over 150 years since his famous book was published, we've actually been able to support a lot of his theories in much more detail than he would have even dreamed of back in his day. So many of you have heard of DNA testing. This is a context in which genetics comes up repeatedly. In the media, we often see references to it in these particular 3 contexts. Paternity tests, trying to identify, among 2 possible dads, which one is actually the father of the baby. Now, sometimes you can tell because the kid looks far more like one potential father than the other, but you can do a DNA test even if the kid looks very intermediate and you can't really tell. It's often used in the context of criminal investigations, where we look for DNA at a place where a crime was collected, and then I try to identify, in the case of forensics, that are examples and what information do they get from this? Even potential lines out there. Gene increase extensively in this class because it's something that there's issues that you should be aware of. First, let's address the question of where do they get this information? How do they identify health risks associated with genes? Well, many of you see references to disease genes in the news. Here's a few headlines out there. Gene increases diabetes risk, scientists find. What particular position are they? Alleles associated with disease have been genetically mapped, where people have found where these genes are in the genome. What chromosome and position are they? This picture shows you several genes identified from various chromosomes across the genome. So, for example, sickle cell anemia is known to be associated with this little stretch here of chromosome 11. The actual gene is known for that. Cystic fibrosis, there's a factor contributing to it there on chromosome 7. There's a lot of these disease genes that have been mapped to particular parts of the genome. And it's not just disease genes. A lot of other genes have come up in the news, sometimes kind of silly ones. Like, I love the name of this one. The fat gene was found by scientists. That was published in the Times. Gene for left-handedness is found. The gay gene has been found in female mice in Popular Science. Some of these are clearly sensationalized in the media. If you look at the effects of fat gene when we Hello, and welcome back to Introduction to Genetics and Evolution. In this video, we'll be talking about some of the very basic history of and understanding of inheritance. And I'm referring specifically in this case to genetic inheritance. Now, if you think back hundreds of years ago, people understood that there was something being passed on from parents to offspring. This This was true long before they knew anything about genes or anything about DNA being the genetic material or anything like that. Well, why is that? How did we know that there's some sort of information being passed on from parents to offspring? Well, the answer is quite simple. If you look around, people tend to look like their parents or their kids. So familial resemblance is obvious, that we all the time see people say, oh, you look far more like your kids than you do relative to somebody else. Now, some of that could be that you're in a common environment. You live in the same house or in the same community as your parents do. However, we don't see as much resemblance in families when you have adoptees. We might have some, but quite often you can have adopted kids that don't look at all similar to their parents. They also may have very little correlation in other traits such as dietary preferences or IQ, which is the intelligence coefficient, things like that. So how did people try to explain this? Well, Charles Darwin, attributed this familial resemblance to some unknown factor he referred to as a gemmule. And there's a quote here, as you see on this slide, from a letter to Francis Galton. If I ask myself how you derive and where you place the innumerable gemmules contained within the spermatozoa formed by a male animal during its whole life, he said male, I cannot answer myself. Now, actually, there was some information at this point about genetic inheritance from the famous pea experiments by Gregor Mendel. However, Darwin did not know about these. Now, it's important to understand genetics, and it would have been actually very helpful for Darwin to understand genetics to help us understand the process of evolution as well. There's something that comes out if you don't really understand genetics that becomes a problem for evolution. So let's look at this. Imagine that you had some tall individuals in a population and some short individuals in a population. And let's say that offspring were always intermediate to the two parents. So if you have a tall parent, let's say somebody who's 6 foot, let's say a 6 foot tall mom and a 5 foot tall dad, would you be 5'6"? Would you be exactly in the middle? Well, let's hypothesize that that was true for a minute. Or as in this picture right here, let's imagine that white flowers and red flowers gave rise to pink flowers. What would happen if this happened over time, over and over again? Eventually, everybody in the population is going to come back to some sort of average. Basically, you'll lose all variation in the population. This is referred to as the problem of blending inheritance. Let me give you a numerical example just to illustrate this point. So here's the numerical example. Now let's imagine you're looking at height, or any sort of continuous trait. And let's say, for example, you're thinking 5 foot x inches, and the numbers depicted here are the number of inches after 5 feet. So we have here a dad who's 5'4", a mom who's 5'9", dad who's 5'5", mom who's 5'8", dad who's 5'2", mom who's 5'6". Okay, so basically any sort of continuous trait. In this case, we're having it range from 0 to 9. So each of these individuals is going to have a kid, and we're going to assume the kid has an exactly intermediate trait to the two parents. So if the dad is 5'4", mom is 5'9", the kid is 5'6.5" inches. If the dad is 5'8", mom is dad is 5'5", mom is 5'8". Again, the kid is 6.5 inches, etc. So look what's happened between the first generation and the second generation. In the first generation, we had a range from 0 to 9. In the second generation, we've lost a lot of this range. We're now going from 2 to 6.5. So what happens if we iterate this over multiple generations? Well, a lot more variation gets lost. So here I just took this person, put them there, this person, put them there, this one here, this one there, etc., etc. And you see, as we iterate this more, we initially had this range from 2 to 6.5, but as we take more and more averages across the population, the range has now gone down to 2.75 to 6.5 in generation 3, and even further in generation 4 to 2.875 to 5.875. So if we iterate this over and over again an infinite number of times, we will eventually lose all variation in the population. Everybody will be exactly intermediate and the same height. Now, if you don't have variation, then you can't actually have evolution by natural selection, because there's no trait being inherited that allows for greater survival or reproduction. Since everybody's the same, everybody presumably surviving or reproducing the same with respect to whatever it is that they're inheriting. But more fundamentally, since when we look in any natural population, we do see variation, and we do see variation that appears to be heritable, this poses a problem for understanding heredity more generally. Now, one possible solution to this was tossed out. This was from Jean-Baptiste Lamarck. So this idea that variation in some way needs to be created or renewed within populations for evolution to happen. So what Lamarck suggested is something along the lines of what's called the inheritance of acquired character. Inheritance of acquired character. Now this right here is a quote from Lamarck's writing. The idea here is that basically if you use something, it then can grow and then that gets passed on to offspring. So some anecdotal examples would be something like a blacksmith. Blacksmith is using their muscles all the time to work with their metal. They tend to have these big muscles, and if you look, kids of blacksmiths often have these big muscles too. Same sort of thing may have been suggested for giraffes, that in stretching to reach the leaves on the trees, they stretch their necks up, and somehow this use of the neck and stretching it actually gets it to be passed on to the offspring, so they then had offspring that had slightly longer necks. This was the idea that was tossed out. Darwin actually adopted this idea for gemmules in some of his later editions of On the Origin of Species. Now, you may know that this is not exactly correct, and I'll show you one case where I messed up. If you go back to my 10th grade high school class, my teacher, Mr. Bennett, asked some questions. He said, if you were to cut someone's left arm off and they have kids, would they have 1 or 2 arms? That was one question on a quiz. The next question is, if you were to cut someone's left arm off and cut their kid's left arm off, repeating for 20 generations, would the 21st generation kid have 1 or 2 arms? Well, I got the first one correct because I said the answer was 2. Because just because you lose an arm, there's plenty of people who lose an arm and have kids with 2 arms. But somehow I thought maybe this would get passed on if this happened over and over again. And this is much more along the lines of what Lamarck had believed. Now, that is not true. In fact, if you were to cut somebody's left arm off and repeat that for 20 generations, 21st generation kid would still have 2 arms. Now, the reason for this, and the reason that variation is not, in fact, lost, the way I portrayed it in the context of blending inheritance, is because we have particulate inheritance. We are actually transmitted by other parts. Also, genetic diseases can crop up, or just new traits in general. It doesn't have to be disease. Now, all of this was known long before. Now, many species are diploid. This is a very important word for you to know. Diploid means that you have 2 copies of the entire genetic code in your body. Right? You have one gene copy that came from one parent, and you have another gene copy that came from the other parent. Okay? Now, you're not intermediate, but you actually have these 2 distinct, complete sets of gene copies for every gene in your whole genome. Now, this is true not just for you, but again, for most animals, many plants and fungi. And in fact, many of the same proteins and structures are involved in inheritance in you as in various plants and fungi, and as well as other animals. And it's because of this that people tend to use model organisms to understand inheritance. When I say model organisms, these are animals or plants that people work with, or fungi as well, that people work with to try to understand genetics. But we're not necessarily interested in them themselves, but we're using them to understand genetics more broadly because we presume that it applies very similarly across all species. So many of you know, for example, the famous fruit fly. People don't work on fruit flies because they love fruit flies or especially care about fruit flies or want to kill fruit flies. They use it because they're easy to work with in the laboratory. Same sorts of things for mouse, for xenopus frogs, for zebrafish, for this plant right here is called Arabidopsis thaliana. So this is why people use model organisms, because patterns of inheritance and the genes involved in inheritance tend to be the same across most living species. Now, let me just give you an idea of what's to come. Foreshadowing. So these are some questions to be thinking about. I'm not asking you them just now. So how do offspring get the genes from their parents? Basically, how do these single copies of all the genes in the genome from your dad and from your mom come together and fuse into what becomes your genome? And are traits controlled by one gene alone? Is there a single eye color gene? Is there a single diabetes gene? Is there a single cancer gene? Things like that. Or are there many genes that contribute to these traits? You can probably guess the answer to that one is the second. And finally, and this is again diabetes gene. Is there a single cancer gene? Things like that. Or are there many genes that contribute to these traits? You can probably guess the answer to that one is the second. And finally, and this is again what I hope to be getting to you by the end of the genetics section, how do we find genes associated with traits? So we'll come back to these points, and I hope you'll join us. Thank you very much. Hello, and welcome back to Introduction to Genetics and Evolution. I decided to add this extra short video because several people expressed that they didn't know a little bit about the scales of organization of genetic data. Now we talk about genes and we talk about how they affect a phenotype, and we talk about how they're hereditary. We mentioned DNA, but we never really said anything about how is it that the DNA sequence encoded in genes actually affects phenotypes. That could easily be the topic for an entire course. In fact, it is a topic for many courses. What I'll do is introduce just some very basic concepts just to help you understand the context of the class that we're doing right now on genetics and evolution. Now, how is genetic information translated to phenotype? Now, we've already talked about how DNA is the primary hereditary material. That is the material that is organized in chromosomes, that is passed from parents to offspring, that is allowing this mechanism for heredity. Now, there are sections within chromosomes that have specific instructions, and these sections are referred to as genes. This is a term that you hear all the time. Now, the question I'm posing to you now is what or how does this impact the phenotype? How is it the DNA sequence that's present in these different genes actually changes your hair color, or your eye color, or your personality, or your IQ? Well, a lot of this, not all of it, a lot of this is done through protein. Now, you hear the term proteins all the time in the context of your diet. Are you getting enough protein? Oh, this kind of food is high in protein. And in fact, proteins are vitally important to many, many aspects of your health, well-being, and development. Some proteins are structural. They're basically putting you together. Some are mechanical. They're essential proteins in various of your muscles, for example. Some of them are biochemical. The enzymes that you use for digesting your food, for example, are largely protein-based. And they can even affect things like cell signaling. So your hormones, for example, have protein components. Now, the central dogma of molecular biology, this is a fundamental principle. And this is an oversimplification, I should stress ahead of time, but it is nonetheless a fundamental principle of molecular biology, is that there are three primary levels of genetic information. They are also interrelated to each other. Now, DNA, as we've talked about, is that hereditary material. It is passed to offspring, but it also replicates itself, both inside your body and in the context of transmitting this genetic information to offspring. So DNA replicates itself. DNA also produces RNAs, and we'll focus on one particular kind of RNA here, referred to as messenger RNA. Messenger RNA is an intermediate between DNA and proteins. There are some forms of RNA that are directly functional, but again, we'll be emphasizing it in this video, the messenger RNA, those ones which are actually putting together the protein sequence. And finally, these messenger RNAs are translated into protein. Now, the structure and content of these proteins are what directly impacts your phenotype, or how you look, how you feel, how you act. Now, proteins are actually composed of chains of amino acids. Again, you've probably heard the term amino acids in the context of things you want to get particular kinds of in your diet. But if you look over here on the right, we have a string of amino acids. This would be part of a protein. If we zoom in on some of them, we see particular amino acids that you may have heard of: phenylalanine, leucine, serine, etc. Now, how is it we get from DNA to these proteins, aside from having this RNA intermediate? Let's talk about each step just very briefly. So the first step is DNA forming this RNA, specifically this messenger RNA. Well, the formation of messenger RNA from DNA is referred to as transcription. Okay? Now, as you may remember from other courses, DNA is double-stranded, and you have this complementary pairing. So for DNA to DNA, you always have these four different types: A, C, G, and T. These are the four different nucleotides of DNA. A always binds with T, C always binds with G, G always binds to C, and T always binds to A. And you see that over here. So here's DNA to DNA. A to T, A to T, T to A, C to G, G to C. Okay. Now, when you're producing the messenger RNA, you have the same sort of complementary relationship, except in this case, the T is replaced with a U. So this is a uracil. So in addition to being a different sort of compound, the DNA is deoxyribonucleic acid. RNA is just ribonucleic acid. It doesn't have the deoxy facet to it. You see it has the same sort of complementary pairing. So we have a double-stranded DNA that has been separated out, and we have this RNA transcript that is being produced in the middle, and you have the same sort of relationship of nucleotides. So you're essentially making a copy of the DNA as RNA. You note the sequence here of this RNA transcript exactly matches the one here at the bottom, aside from this switch from T to U. See TAC, TGCC, UAC, UGCC, etc. So this is how the DNA is making its copy as messenger RNA. Now what happens is this RNA copy of the gene sequence